So thanks for coming here and listen to what I want to say. So I would like to tell you about geroscience, which is inspired. But more than anything, since I've been in France for the last 2.5 months or something like that, I've seen that people don't understand the difference between geroscience and aging biology, both of which interest me, by the way, but it's not the same thing. Some of you might recognize these guys. These are the Beatles. In 1964, they put out a song called 8 Days a Week. They were having so much fun that they wanted to have more fun. So they wanted to have 8 days a week. That was in 1964. We're in 2020. So as you can see, both in France and the States, we have increased our lifespan. Lifespan is increasing dramatically all over the world. We have increased it from 70 to 80 years. So we do have 8 days a week. Not bad. We have 8 days a week now on average, right? The question is, are we having fun? And there, unfortunately, I have to say the answer is no, not so much. Why? There has been an increase in life expectancy. This is data from Eileen Crimmins showing that there is an increase in lifespan over this period of years, 8 years in there. The problem is that this increase in lifespan is dwarfed by the increase in diseases. So, are we having fun? No, not really. We're living longer, but sicker. That was not the idea. That was not the goal. So, what happens is that it's because we focus mainly on death rather than on health. So, this represents any individual which is born because of genes, environment, with a certain capacity on each one of its vital symptoms to last for a while and then eventually go down. This is what happens, but each one of us is different. So in this case, this person will die when the first system goes out of steam. In this case, cardiovascular disease. Okay? So what does the scientific community do? War to cardiovascular disease. Cardiovascular disease is killing us. Okay? So what if we succeed? And with statins, we have succeeded to an extent. We extend. Well, okay, so we forget there's other diseases going on. We focus only on those that kill us. So, with cardiovascular disease, okay, we're gonna that particular individual, not as a community, but that particular individual will live longer because cardiovascular disease did not kill it. The problem is that living longer with a severe case of dementia, sarcopenia, cancer, immune functions, etc. A lot of things are happening at that time of age. So curing one disease, cancer, cardiovascular, is not sufficient. It's not the end. So what happens is that the world is aging very rapidly. So in this graph, you see that there's right now in 2020, there's more people age 65 than people under 5. This is worldwide, not just in France, not in the States, it's worldwide. And we assume that this is a big problem. It's a big problem because we're going to have more health issues. We're going to have a diminished workforce. So there's social and health issues that are coming up because of this trend of increasing lifespan. So we think it's a big problem. The question is it really a big problem? Because I tend to be a very optimistic person in general. I tend to look at the sunny side of things. So is it really a big problem? Unfortunately, in this case, my optimism is not enough. Yes, it is. It is a big problem, a huge problem. And the reason is because if we don't change the way we're addressing health and disease, we're not going to surmount this problem. So in the 20th century, we had a big success. That's how our life expectancy has increased. But the major problems we had were diseases of simple etiology. We had bacteria, we had viruses, things that we were able to conquer through sanitation, vaccines, public health services, etc. This approach will not work against the chronic diseases that we're dealing with. Cancer, cardiovascular, diabetes are not simple diseases caused by an external attacker. They are actually our own. So the major risk factor for chronic diseases is aging itself. So the enemy that we're trying to fight is ourselves. It's our own body that we have to address. That's why it won't work the same way we did before, because it's a completely different battle. So, let me switch a little bit to why did I come here to Toulouse. I was interested in the project Inspire. Well, I was looking for a challenge. That's the bottom line. But I was interested in Inspire for a very simple reason, which is here. Inspire focuses on the drivers that maintain our health. So basically, classical medicine focuses on the negative things. We focus on disease. We focus on comorbidities, on dependency, all of those bad things. Okay? Those are unhealthy things. Inspire focuses on the things that keep us from getting there, on the positive side. How do we avoid being frail? How do we increase our resilience? How do we increase our intrinsic capacity? So that's a healthy side of the equation. Now, how they do it, they have a clinical translation and basic aspects to it, and the important thing is to try to make them work together. And I'll get back to this in a few minutes. The question that came to me is because actually in the project, they kept talking about geroscience, which was exciting to me to hear people in France talking about geroscience, something I had created. But very soon I realized, hey, these people are not doing geroscience. Okay, so why are they not doing geroscience? What is the difference between geroscience and aging biology? And to do that, I had to define both. So what is aging biology? Aging biology is a description of what happens with age. And just to make clear that I have nothing against aging biology research. There is a point. It's too weak, that's what it is. So this is data from my own lab many, many years ago. I found this gene that goes up with age in 3 different strains of rats. Okay, it's descriptive. Then I spent a lot of time doing mechanistic studies on how it goes up and what's the consequences and so on. That's aging biology. It's a description of something that happens during aging. It's also a description of disease susceptibility. So here we are at the cancer center. This image to me is amazing. These cancers are all on different tissues of different genetic drivers. Everything's different about them. However, the increase, it really doesn't have that battery. The increase as a function of age is the same for all of them. It's more than that. It increases around 60, 70 years old. That's when the big jump happens. That's about two-thirds of our lifespan. It happens to humans, it happens to mice, it happens to dogs, it happens to elephants. They develop the disease at different times because they had different lifespans, right? But it's always about two-thirds of their lifespan, okay? So basically a mouse, a C57 black 6 mouse, will develop cancer, 80% of cases, okay? It will develop it when it's about 20 months of age or something like that. So different species or different individuals will have the development of these diseases more or less. Some of them will develop, some of them will not. And that's determined by your genes and your environment. What aging does is it determines when you get it. If you're aging well, you're going to develop your disease for which you have susceptibility a little bit later. If you're aging bad, you're going to get it a little bit earlier, okay? The same is true for Alzheimer's. And what I want to point in here is that these curves are not linear. They're a sudden rise at the end, both of them. That means that it's not simply the accumulation of damage. It's not accumulation of mutations, as many cancer people think. It's not just that. It's not accumulation of A beta. You can be born with mutations in your APP gene, your presenilin genes. You will get Alzheimer's. You will. However, you're not going to get it when you're a toddler. You're not going to get it when you're young because you're resilient. You're going to fight that accumulations of protein that you're being produced as faulty because of your genes, you're going to remove it, and you're not going to accumulate it. It's only when you lose the ability to defend yourself that you start getting the problems with the disease. So really, aging is not just the passage of time, it's your loss of ability. And that is the important point to remember here. So it's a description of something that happens with aging, a description of disease susceptibility, a description of how you respond to the environment. This is an example with coronaviruses. You're looking at the lung of monkeys, actually. The control, nice alveoli, nice looking. If you infect with a virus a young or an old monkey, you see that there's a difference. Okay, so that's again a description of something that happens with aging. In this case, responsiveness to a virus. So aging biology is descriptive and mechanistic. Okay? What have we learned through all of these studies? So aging biology, when I joined the field about 30 years ago, it was the most backward field you can imagine. Nothing interesting was happening, which is what attracted me to the field. I thought, hey, I can do something here. I can help. I can make a point, right? So what we have learned is that, first of all, that there are genes that control longevity. Okay? On your left is the classical DAF2 mutation that increases lifespan in C. elegans by about 40-50%. On the right is an extreme example where you increase the lifespan of C. elegans by about 10-fold. So if we extrapolate to humans, that would be living to 800 years. It's not going to happen. But in the worms, we can do it. So we have found in mice too, and there's many, many genes. And this is an image that I got about 10 years ago. So it's many more now. Okay? So there's many genetic ways of affecting longevity. There's also behavioral ways. Caloric restriction is the best known. You eat about 40% less than what you want, and you live about 40% longer. And it feels like you live forever, but it's because you're not eating properly. Then we have pharmacological ways, like rapamycin. It increases lifespan about 20%. That's not a little thing. 80% of 80 years is 16 years. If we can increase our lifespan from 80 to 96, that would be quite something. So basically, the bottom line is that aging is malleable. It's much more malleable than we thought. So you can have behavioral things, eating less, genetic. By the way, there's about 800 genes that have been described that extend lifespan. We don't look at things that shorten lifespan because that's very easy to do. But they extend lifespan, about 800 of them. We have pharmacological means. So it's much more malleable than we thought. People always thought that aging, you cannot change it, and that is true for your chronological age. The important thing, however, is I've been talking about lifespan. The important thing is Healthspan is also improved. And there's many, many hundreds and thousands of papers that show this, that these manipulations that I mentioned lead to an improvement in Healthspan. Not on every aspect of Healthspan, and that's something that we have to keep in mind, but it improves Healthspan in general. Rather than give you a list of things, I will show you a movie. These are mice that are 38 months old. These are centenarian mice. These are very, very, very old mice. Okay? The ones on the bottom are behaving as you expect centenarians to behave. They don't look very good. One of them has a cancer, a tumor sticking on the side, etc. The ones on top are the same age, and they're acting as if they were kids. They're exploring their cage, they're rearing, they're looking around. They are the same age, but they have been 6 months on rapamycin, which is one of the drugs that we have found that increases lifespan, and in this case, Healthspan as well. So, what have we found really? Is that we really understand pretty much at the molecular and cellular level what are the main drivers of aging. We don't know them all, that's for sure. In fact, most of you probably have seen the one on the right, which is a paper by Lopez Otin. I prefer the one on the left because it's mine. But you will see that in here it says other biology. We don't know everything. No, we don't. We don't. But the main drivers, we know them. Okay? And that is what's allowing us now to start thinking, okay, if we understand the process, at least in a rudimentary way, if we understand it enough, we should be able to apply this to improve the lives of humans, dogs, whatever we want. So that's how we go from basic biology to geroscience. That's what geroscience is, and I will explain that. So the problem that we have is that biomedicine really focuses on death and not on health, as I showed you before. We focus on what kills us. We put a lot of effort, and sorry about cancer, we put a lot of effort on cancer and cardiovascular disease, rightly so, because it kills us. But we're forgetting to put enough money in arthritis, infatigability, on those things that make our life miserable without killing us, right? The focus is on death. And that's what geriatrics and geriatricians and gerontologists do. They focus on the diseases and try to prevent those diseases kill us. I would say we have to go a little further and look at how aging leads to the diseases that lead to the death. So we're still looking at death, but I will qualify that a little bit later. So let me give you an example here. I've been saying that aging is a major risk factor for all diseases. Is that true? If we think about, and I'm talking about cardiovascular in here, not about cancer, the classical risk factors. Well, it's cholesterol, obesity, diabetes, cholesterol, high blood pressure, and cholesterol. Why so much cholesterol? Because we have a drug against it. That's why we think it's so important, right? It's the only one we can control. But let's imagine something. This is me and my daughter. Imagine that I'm a really, really bad father, which I don't think I am, and I take her to eat junk food 3 times a day for several years. That would be possible in the States, very easy. Less easy here, but anyway. Let's imagine I do that. Her risk for cardiovascular disease would indeed increase about 5-fold, which is terrible. That's why I don't do it, right? However, what happens to me is different. It's the same cholesterol. It's the same bad habits. Why do I die? Because I'm old. That's simple. Okay? And it's not, as I showed you, it's not because of the accumulation of cholesterol through my life. Because remember, the accumulation is like this. It's not linear. So yes, I do have more cholesterol in my arteries than she does. Yes, a little bit. But that's not the reason. Let me show it in another way. This is data from the Framingham study. And it shows that if you're 30 to 34 and you have all of these conditions in the worst possible situation, very bad cholesterol, very bad HDL, you're diabetic, you're a smoker, etc., you get 16 negative points. If you are 75 years old and everything is perfect, you get 15 points. So the main risk factor is not cholesterol, it's aging. Okay? So, how do we go from here to doing something about it? Let me just explain. You go to a doctor when you have a disease. Unfortunately, we're not preventative enough. What the doctor says is evidence of one disease or another. He says that you are a little bit demented, you have very bad memory, and you don't recognize your parents or your children. Chances are you have Alzheimer's. Or you have you had a heart attack. Okay, you have cardiovascular problem. Those are the specific physiopathology of that disease that defines that disease. But if we think about the reason we get that disease, the main risk factor is aging. And then there's the physiopathology. It is the APP, it is the cholesterol, but it's a minor component, aging being the major one. And more important than that, aging is a common component for both of these diseases. So what I'm proposing is that we should be focused on aging rather than the specific diseases. And this is what gave rise to the concept of geroscience. Basically, we have enough knowledge about the biology of disease. We have enough knowledge about biology of aging. Let's combine these things and propose geroscience, where aging biology is at the center not only of one disease, but of multiple diseases. So the corollary to that, which is the real Geroscience hypothesis, is that if we accept that aging is at the center of all of these diseases that are put in there, if we can reduce the rate of aging, as we could do it in the mice with rapamycin, you saw that, we're going to reduce the rate of all the diseases all at the same time. Therefore, we're going to increase health as opposed to curing a disease. And this is not only true for these diseases, it's also true for things, as I mentioned before, frailty, fatigability, hearing loss, all of those things that we don't pay much attention to, but are also age-related. They're also driven by the process of aging. So we're going to improve the health of the population. So this is the concept. And let me put, again, I come back to Alzheimer's for a political reason at the NIA, at the National Institute on Aging, which is where I also work. If we want to diagnose whether a person has Alzheimer's or not, yes, we have to look at the brain. We have to look at the function of the brain. We have to look at amyloids, tau, whatever. We have to look at the brain if we want to determine if a person has the disease. However, if we accept that aging is the driver, the main driver for the disease, and aging is affecting all your body, every organ in your body, at different rates perhaps, but it is affecting every organ in your body, then in principle, and this is a theory, it's a hypothesis to be tested, in principle you should be able to diagnose your risk of developing Alzheimer's disease by looking at the cells in your belly button or your toes, because those cells are aging at the same rate, or in parallel, let's say, not necessarily at the same rate, but in parallel with the cells in your brain. So if your cells in your belly button are showing signs of being aged, chances are you're going to have diseases of aging. So how do we do that? Okay, let's say for Alzheimer's, we say most likely culprits involve the stress response and proteostasis. If we had measures, and we don't, if we had ways to measure all of these pillars of aging, we could determine for any given person its level of risk for different things, okay, for each one of these pillars of aging. And based on that, plus the role of each one on the disease in question, we should be able to say, what is your relative risk? Now, this is dreaming. Okay, I'll be the first one to say. We're not even close to this. But this is the concept, this is the idea, this is where I would like for things to go. So this is the concept. Let's see what are the facts. Do we have anything going in this direction? Actually, we do, quite a number of studies. In here we have a work from Marco De Maria and Judy Campise's lab, where they use a genetically engineered mouse where you can actually remove senescent cells by giving them ganciclovir. And you can see, focus on the activity. If you give the mouse doxorubicin, the orange circles, the activity goes down. Yes, when we give chemotherapy to humans, they get fatigued. They don't want to move, right? However, if at the same time you give the doxorubicin in the last set of data, at the same time you remove the senescent cells by ganciclovir, well, the mouse moves as much as the control. It's not fatigued in spite of having doxorubicin. That's quite amazing. In fact, in a previous study to this one, they had shown that there was a correlation, and again, this is a correlation, nothing, no cause effect. A correlation that people who had more senescent cells suffered more fatigue after chemotherapy than the ones that had less. So in humans, seems to be at least a correlation. There was also a decrease in the number of tumors, but that was not a point of the experiment. Not that it's not important, of course, but at least the reason they measured it was because they had to make sure that was not the opposite. That, yeah, you're less fatigued, but the chemotherapy didn't help you. You have more tumors. No, it was not the case. Another example, so this time using senolytics, not a genetic model, but senolytics, so drugs that kill senescent cells preferentially. They did a study in humans with idiopathic pulmonary fibrosis. The reason I put there may improve is because the study is only 14 people without controls. So very, very, very preliminary. Nevertheless, the preliminary result shows in here, you're probably not going to be able to see, but in red, I highlight the physical functions. There's a decrease, an improvement in physical function that's statistically significant in all of these functions, 4-meter gait speed, grip strength, etc. There was no direct effect on pulmonary function, because that's not what we're attacking. We're not attacking pulmonary function. We're attacking aging. Okay? So there is evidence that the hypothesis is true. Another set of examples, rapamycin, that's another one of the drugs that we're very interested in studying. This study was done in mice and dogs. And when I talk about dogs, it's your pet dog, the dog that you have at home, a real animal, not a pseudo animal. It's not a genetically modified or laboratory bred animal. It's your dog, the one that licks you and shares its microbiome with you. So what they did, also they gave them rapamycin for, I don't remember how long, unfortunately. And they look at cardiac function. The reason they look at cardiac function is because that's what this group does for a living. So the results in blue and red are in mice, in green and purple are in dogs, and what you can see for 3 different cardiac functions, over the time period of the experiment, there was a decrease in function because the animals were aging, right? However, during the same period of time, the animals with rapamycin had an improvement in all 3 of the functions. So pretty good, considering that rapamycin that we know has no direct effect on the heart. The next example is even worse in that sense. It's in humans. It's a work of Joan Manick from Restorbio. What she did was she gave rapamycin as well as another TOR inhibitor to elderly people right before, or several weeks and months before they got their yearly influenza vaccine. 3 months later, she measured the levels of antibodies against 3 epitopes of influenza vaccine. The blue line shows what she was hoping to get, a 20% improvement. And if you see in the last set of bars, what you get is a 30% improvement in the response to the vaccine. 2 things to notice in here. There's no other treatment that improves the response to vaccines to this level. When we're talking about improving response to the vaccine, we're talking about 5%. This is 30%. This is much more serious. The other thing to notice is that the treatment that she chose is rapamycin. Rapamycin is an immune suppressor used in the clinic as an immune suppressor. The outcome in this experiment was the immune response. So you get an improved immune response with an immunosuppressor. Pretty cool. The reason is simply because you were not acting directly on the immune system, you were acting on the body as a whole, improving health as the individual is getting old. So there is some positive results coming up. This is all still being done. So I'd like to go into how do we develop geroscience in Inspire and beyond, because that's what I came here to do. My goal is very simple. I want to make Toulouse the center for geroscience in Europe. That's simple. So how do I get there? The main strengths that we have between Inspire and geroscience is that they're both focusing on the same. They're focusing on not on specific diseases, but on prevention of frailty and maintenance of intrinsic capacity. So the focus is on health. And that to me is very, very important. Focusing on health and not on disease. Now, in practical terms, what we have strong is that we have two parallel cohorts of humans. A 10-year longitudinal cohort is very good because they're following every 4 months. That's a very, very deep phenotyping, really. And we have animal models, mice, dogs, and fish. So there are things that are positive. Unfortunately, there's also some weaknesses. And this is going to be my job to try to change it. There's limited aging biology expertise, and consequently, minimal to no aging biology networking. Most people in here don't even know the people that I worked with in the last 30 years. Okay? And scientific, there's not enough samples, there's limited animal cohorts, so there's things that we have to work with. But what I want to emphasize is, as a result of these weaknesses, because people don't know aging biology yet, what we're doing is actually aging biology and not yet geroscience. So anybody who's telling you that they're doing geroscience, they don't understand the difference. There's a lot of interesting biology that can be done. One thing that I always have to point out is, to me, there's absolutely zero goal, zero value on measuring something that we already know to see if it's also true in Occitanie. I don't care. Yes, IL-6 is going to go out with age. Yes, it will, like it does everywhere in the world. Now, the value, however, of measuring this type of things is that in here we're not comparing with age, but does it correlate with frailty? Does it correlate with intrinsic capacity? Very few labs in the world are doing that aspect. Okay, there's a few, but not that many. And I think there's a lot of omics that can be done. Now, this is still descriptive, and that's why it's biology of aging, it's not geroscience. But I also want to emphasize again, I have nothing against aging biology. I was the director of the division of aging biology. I was not the director of the division of geroscience because that doesn't exist. I've done aging biology all my life. Okay, so I'm not insulting anybody by saying, no, you're not doing geroscience. No, that's where I want you to go, but nothing wrong with starting with that. There is a unique opportunity for Inspire that I want to pursue. We can measure molecular resilience in relation to frailty and intrinsic capacities. What do I mean by that? We focus a lot of our research in geriatrics on the concept of frailty, primarily pushed by Linda Fried. The problem is that frailty happens way too late. By the time my physician tells me that I'm frail, what he's telling me is, Felipe, you're about to die. I'm not interested in that. I will know it by the way, right? So I also make the point that frailty happens very late. I am not frail at all, right? Not at all. However, my resilience is not the same that it was 30 years ago. When I was 30 years old, I could party all night. Now I can't. Or I can, but I cannot recover after. So resilience is something that you lose early. And therefore, there's much more value if we can measure resilience than if we can measure frailty. When we measure resilience, if I am told that for my age, my resilience is a little bit low or is a little bit high, that will tell me what I have to do before I get diseases. So there's a value on trying to do that. So basically, we focus a lot on frailty and disease and how those things lead to death. I'm proposing that resilience is upstream. Resilience is what leads to frailty, leads to disease, and all of these things, unfortunately, interact with each other in a negative loop. But the entry point, I think, is a loss of resilience. So when we try to join aging with disease, we're focusing on frailty, and I think that we should add another piece of the puzzle, resilience. So what can we do in Inspire? Inspire focuses on the measurement of intrinsic capacity, which is defined by the World Health Organization as the ability to do the things that you want to do. It's not disease. It's the ability to do the things that you want to do. I play the guitar and I paint. If I get arthritis on my fingers, I'm going to be a very unhappy person, right? It's the things I want to do. But the important thing is that it's intrinsic capacity, the ability to do it when you're challenged to do it. So that's the same as resilience, okay? Now, the current definition of intrinsic capacity by the World Health Organization is physiological. Psychological, cognition, locomotion, sensory, etc. is physiological. If we want to develop drugs or treatments for it, we need to define it molecularly. And that's the question I would like to address. Can we measure it at the molecular level so as to identify potential targets? And I think we have a good opportunity to do it in INSPIRE. We don't know how to measure resilience. We really don't. In humans, not at all. And in humans, we cannot really stress them very easily. Even in mice, we don't know how to measure it. We're trying to do that. But in here, I have my own personal definition. What I'm talking about when I talk about molecular resilience is this. It's the capacity of every cell in the organism to respond to a challenge. So it's not unique to the cardiovascular disease or the heart or, no. It's every cell. A good example is chemotherapy. When you give chemotherapy to a person, every cell in the body has to up its resistance to DNA damage. Or if not, they will die. So how is that capacity changed with age? Can we measure that? Okay? And when I started this project in the US, I insisted that it had to be simple. If you need a 7 Tesla machine to do it, I was not interested. So very simple things. How do you recover from anesthesia, from chemotherapy, from radiation, from trauma surgery, starvation, etc., etc. Simple things. But we don't know how to do it yet. This is what we're trying to do. The most crucial thing to take advantage of the INSPIRE project would be to ask if we can measure molecular resilience in the cells from the donor, right? Because we cannot stress the old person. But can we take fibroblasts from them, stress those cells, and does it reflect the resilience of the donor? Maybe not. We have to see. There's suggestion that it does work, like this work from Rich Miller's lab, where he showed a mouse strain that lives longer, the dwarf mice. When you test them when they're young, they have better resilience than the average mouse, than the control mice. Okay, so in principle, you can measure it. He measured in fibroblasts. Now, the limitation is that this is a genetic manipulation. So, of course, the genetic characteristics were maintained in the cells in culture, right? But it does show that even in fibroblasts, you see the difference. The question is, can we do it in real life when the differences are not going to be as dramatic as with this genetic manipulation? So the idea is you take a population, the INSPIRE cohort, you measure whatever you want to measure, resistant to chemotherapy. You're going to get the extremes of the curve, the ones that react particularly well, the ones that react particularly bad. And since the cohort is, oops, that's not what I meant. Okay, since the cohort is translational, longitudinal, you can see as a function of time, do these individuals that are in the extremes show a difference in their development of frailty and other symptoms? The important thing is to do it in the cells in culture, and we just don't know. So I can't promise you that this is going to work. It's a research project that I want to launch. So let me just finish, and this will take me only a few minutes. What I think is needed for geroscience in INSPIRE. And these are basically they are the specific aims of the grant that I'm writing to myself. Basically, these are the things that I would like to do in Toulouse. We need to collect human samples of value to biologists, and I'll tell you what, and then I will go one slide per issue. Develop better collaborative projects in geroscience, expand and sustain the animal resources, develop new curriculums, new research plans, and develop new geroscience-based clinical trials. It's a tall order. It's a lot of things. Let's go one by one with one slide each. Collect human samples of value to biologists. There has to be a crosstalk between the clinicians and the biologists, so that the samples that are collected are the samples that will help the biologists. As I mentioned, I don't care about measuring things in blood that we know that change, or that are static and they produce a correlation. To me, personally, I think that the obtaining cells that we can challenge and measure things like resilience is much more important. Where do we obtain the cells? How many cells that we're going to get? How we're going to handle them? This is something that has to be a cross collaboration between clinicians and biologists. We need to develop collaborative projects in geroscience. So I have in here the heads of several INSAN groups and CNRS groups, whatever, in Toulouse, and I put them within the context of the pillars of aging. So we're covering several of these things. Okay, pretty good. But one of the things I like about my depiction of the pillars of aging is the emphasis on the interactions. We need to have interactions. We cannot have a situation where this institute is doing this and this institute is doing that. No, we have to cross-talk. And that is something that's not happening so far, and it's going to be my next job, basically, to work on that. And the question there is, what can you guys contribute? Where do you fit? Some of you will fit in different aspects, and we need to learn about that. Expand and sustain the animal resource. I think that Inspire did the right thing in choosing the cohorts. The killifish and the pet dog are the hottest models in aging biology right now. The killifish because it's the shortest lived vertebrate we know of. Okay, lives only about 6 months or 5 months because it lives in ponds that desiccate during the summer. So it has to produce all its life and lay eggs before the pond dries. Okay, and the interesting thing is that they age with the same problems that we do. So it's a fantastic model. The pet dog, I already mentioned, they share everything with us. They share our socioeconomic status. They share our microbiome because they lick us all the time. They share our health care, everything. They live with us. So to me, they're a better model of human than, say, non-human primates that are always hailed as the best model because of their genetic similarity with us. Yeah, they might have this genetic similarity, but they don't share their environment with us. So to me, the pet dog is a fantastic model. And the mice, of course, because everybody uses mice. I don't like mice much because we have forced the mice to do things that they normally don't. Okay? We have forced them to develop something that we call Alzheimer's. It's not Alzheimer's whatsoever. Mice don't develop Alzheimer's. You can't force Alzheimer's into a mouse. It doesn't work. The model is awful. Okay? But everybody uses mice. So we have to use mice too. So we have to expand that colony a little bit. This is a very exciting thing. Felipe has this project to develop. This is a master's program. There's a companion Ph.D. program. So basically what it is they take students of different origins that come from the hard sciences, mathematics and physics and so on, from the pharmacy, medicine, etc. They take them from different areas and they have them together learning about each other. So at the end, the student still has a focus, say, on physics, but understands the biology. That's what we need. So I think that's a fantastic thing that I won't have to work on. I just have to support it. And last, we want to eventually develop a geroscience-based clinical trial. So what's the concept of geroscience is that if we take we call them geroprotector, a drug that affects aging process, we're going to affect multiple diseases. So there's this same trial that's being done in the States, where what they're measuring with people who are already lipid sick, so that they develop the diseases faster, the time to any of a panel of the different diseases. If we treat them with metformin, are we going to delay the appearance of the next disease? That's the bottom line. I think we can do a similar thing, but switching the primary and secondary outcomes. We can look at the functional outcome. What happens with intrinsic capacity? What happens with frailty? Can we delay those by treating with a drug, whatever drug we want? So that would be a different way of doing the same thing, but again, focusing on function, on health, and not on disease. If you will allow me, I have 2 or 3 more slides that I want to share with you, because we tend not to think about this. Health is much more than the absence of disease. I've been telling you all along that I care about health and not about disease. Health is much more. So the bottom line is that we can address the health issues. But health also requires well-being, affection, self-worth, acceptance, happiness, economics. All of those things that we are not giving our elderly right now. So basically, this is your life. In short, you are born, you are a child, then you train until you're 25, a little bit more sometimes. Then you produce and consume. Then you retire at 65, you become frail, and you die. That's it. Okay? However, because of the advances that we're having a lot of people living much longer in good health. So what happens is that not everybody dies at 80. Some of them will become frail early, and we have to continue taking care of those. I'm not saying that we should not do that. Of course we should do that. But what we are neglecting as health professionals is the increasing number of people who are at the opposite side. Because we are living longer, we have all of these very resilient people, very healthy people that are willing to participate in life, and we're not letting them. I don't think that's feasible. If we increase our lifespan, say, to 90, to 100, we're going to leave these people sitting in front of the TV for 40 years? No. So basically, we have to rethink our things. Maybe we continue our life the same. Changing the retirement age is politically not going to happen. But at that point, you retrain yourself, you do something so that you keep being productive. Now, society has to accept that. Interestingly, I made this figure about 4 or 5 years ago. And interestingly, I'm happy to say that that's exactly what I'm doing. I retired at 66. And instead of going to watch TV, I came to Toulouse to challenge myself. This is what we have to do in a larger scale. So health is not everything. We also need the social and economic aspects. And that is the entire thing. And that's what we have to strive for. And that's it. Thank you.